So radiofrequency radiation is emitted from cell phones. This was nominated to us. I'm too tall. All right. So cell phone radiofrequency radiation was nominated to the NTP program in 1999 from the U.S. Food and Drug Administration based on several reasons, mostly because of the widespread human exposure, and that's actually been increasing exponentially, it seems like, over the years since this was nominated back in 1999. Currently, I just looked at some information this morning, and it seems that there's more wireless subscriptions than there are people in the United States. There's about 318 million people in the United States, and about 378 million subscriptions. Last year, Americans talked on the phone 2.8 trillion minutes. So if you divide that out, that's about 145 hours per individual in the United States. So worldwide, this exposure is estimated to be over 7 billion people. One of the reasons for the nomination was also that at the time, and even currently, there's very little known about the health effects of long-term exposure in humans to cell phone radiofrequency radiation. And the current guidelines, which have been around for a number of years, are based solely on protection from acute injuries, which we know are caused by thermal damage. And we know that human exposure actually occurs below this thermal level in what's sometimes referred to as the non-thermal level. So specific concerns for cell phone radiofrequency radiation obviously is the head, because the wireless devices are used in close proximity to the head and the brain. So a lot of the studies that have looked at cell phone radiofrequency radiation have focused on this. Epidemiology studies have looked at looking at the association between the potential increase of tumors in all parts of the body, but primarily in the head. And what they've demonstrated is that there is a potential increase in glial cell tumors in the brain and vestibular schwannomas, which are also referred to as acoustic neuromas, and that these tumors may be associated with exposure to cell phone usage. However, a lot of these epidemiology studies have some inconsistent results. There are some studies that show differing results from some of the studies that are positive. There's a lot of confounding factors in these studies and biases. When you ask somebody how long they talked on their cell phone 10 years ago, I don't know what I had for lunch last week, no less how long I talked on my phone back in 2005. There's a long latency period for tumors. We're not sure that we've reached that period yet, so we're not sure how the results, that may be affecting the results of the epidemiology studies. Studies in laboratory animals have a lot of inconsistencies and inadequacies in their studies as far as limitations in their designs. A lot of the studies look specifically at the brain, but didn't characterize exposures to the rest of the body. Some of the studies use short or very low exposures. And then there's just a lot of physical limitations and logistical challenges involved with testing something such as radiofrequency radiation. In 2011, the IARC convened and reviewed the data that was in the literature, and their conclusion was that cell phone RFR was possibly carcinogenic to humans. That means that there could be a risk associated with exposure. And so when this came to us, we looked at a lot of the studies that were out there already, and many of the animal studies were conducted using this setup that's seen on the right-hand side of the slide. This is often referred to as a Ferris wheel exposure system. As you can see, it looks a lot like a Ferris wheel. In the middle is where the antenna is, and then the animals are exposed in these cones across the outside of this Ferris wheel type chamber. Now, the benefit to this type of exposure is that there is a uniformity to the exposure. The distance that the animal is from the antenna is equal in all the animals. However, it's very stressful on the animals to be contained and restrained. So usually these studies only occurred for about 2 hours a day for the exposures. So early on, we established a collaboration with the National Institutes of Standards and Technology to develop and to look at the exposure systems and develop a new exposure system that would address some of these limitations in previous studies and allow us to design studies that were more robust and allowed us to expose these animals unrestrained in individually housed animals. We wanted the exposures to extend for longer than the 2 hours a day that a lot of these studies were exposing the animals. So we wanted a minimum of about 6 hours a day. We wanted to make sure that the exposures were to a uniform field, much like they were in this type of setup, and that we wanted to make sure that we could expose the animals to the maximum power that we were capable of exposing them to where the animals would still thermoregulate, and this would be in that non-thermal range that I mentioned. We also wanted to make sure that the studies were using technology that was in existence in the United States at the time, and they continue to be. These are the two types of networks that are in the United States. These are considered modulations. One is the CDMA, or the Code Division Multiple Access, and the other is the Global System for Communications. These are primarily used both in the US and Europe. And what NIST had proposed was that we look at an exposure chamber called the reverberation chamber design. And so basically what a reverberation chamber is, it's a shielded room with RF antenna that exposes the animal to RF. In that room there is a vertical and a horizontal paddle that's specially designed at very specific angles, and these spin, and it creates a homogeneous environment within the chamber. So the benefit of this type of exposure system is that the animals are exposed to a uniform field from all directions and all polarizations, and that over time this is averaged out and the fields are homogeneous over the large volume of the chamber. Another benefit to this type of study is that the characterizations of the exposures can be well characterized and monitored in real time. Concurrently, while we were working with NIST, we established a collaboration with NIEHS Foundation, and they created some complex computational simulation models of RF dosimetry. They looked at estimates from these models of whole body exposures, as well as organ-specific internal field strengths, to determine the specific absorption rate, which is the absorption of energy into biological tissues. This is expressed as a watts, or power per body weight, or kilogram. So the SARs are expressed in watts per kilogram. The overall goals for these modeling simulations was to evaluate the SAR, or the exposures within the animal, to determine penetration, to make sure that the animal was exposed not just on the surface, but the exposures were through the entire animal and as uniform as possible to each of the tissues internally. Also was to determine the effect of, or the potential effect of, particular body parts and internal organs. One of the concerns was that the tail itself kind of sticks out from the animal, acts kind of like an antenna. The concern would be that the tail would absorb much of the radio frequency radiation, and obviously the tail is involved in thermoregulation, so that this might affect the animal's ability to thermoregulate, as well as if we had to adjust the exposures based on the tail, we might be underexposing the rest of the body. So these models helped us to look at this issue. Thirdly, it allowed us to evaluate the impact of frequency on SAR in laboratory animals, both the rats and mice. So I don't show any of this, but the results to this are being published. This has been submitted and will be published soon. The final results of that was that the dosimetry modeling basically demonstrated that the optimal exposure frequencies for rats and mice were 900 megahertz for rats and 1900 megahertz for mice. This allowed us to get the maximum penetration of the SAR into the animal without creating hotspots within the animals. So the first thing that we needed to do was to build the facility. NIEHS Foundation first built and tested a prototype in Switzerland based on the parameters that were obtained from the early studies done on the reverberation chamber concept at NIST. We needed to then, once those were tested, we needed to construct 27 reverberation chambers in Switzerland. And the reason that we needed 21 separate reverberation chambers is that each chamber is actually an exposure for a particular power level and a particular modulation. So as you see below here, we had one control chamber that did not have any RF. This chamber was designed the same as all of the other chambers, with the only exception of it not having an RF antenna. And then we had a low, medium, and high GSM chambers, so that's 3 chambers, and a low, medium, and high CDMA chambers, that's 6 chambers. So overall, we needed 7 chambers. That's for the mouse study. When we get to the rats, the weights of rats when we get to 2 years are quite different between males and females. So to make sure that the SARs were appropriate for males and females, we needed to expose them separately since it was based on body weight. So we needed 7 chambers, each of these 7 chambers for the males, and each of these 7 chambers for the females. So when you add that up, that's 21 chambers. Those chambers were shipped to ITRI, or IIT Research Institute in Chicago, the contract lab where these studies were conducted. And at the time, architectural changes needed to be made to the facilities. We have some very interesting pictures that I won't show this time of these chambers being lowered down into a slab in the concrete into the basement of the facilities. So there was quite a bit of work that was done before we could even expose an animal. This is a picture just down one of the hallways of the reverberation chambers. This is one particular chamber. You can see they're kind of lined up here down the hallway. Once they were installed, they needed to have HVAC and electric and everything hooked up so the chambers could function, all the electronics for the exposure system. So again, that was quite a bit of work that went into designing and constructing the facility. On the left, this is a picture of the empty chamber. These are the paddles that I talked about. This is the vertical paddle. This would spin during exposure. This is a horizontal paddle. This would also spin during exposure. And this creates that homogeneous environment within the chamber. You can see on the right, this is with the chamber racks in. These chambers were actually designed only for the footprint to only house the number of animals that we needed. The larger the footprint, the larger the cost. So you can see that we've used every bit of space here within the chamber. So the cell phone studies were conducted in 3 phases. They were conducted in Harlan HSD rats as well as B6C3F1 mice. Primarily, I'll be talking about the rats today. I do have some data from the 5-day thermal pilots for both rats and mice. These were a series of studies that were designed to look particularly at the effect of body temperature to kind of define the non-thermal and thermal range. So as we move subsequently into our next studies, we would select the proper exposures. So in the first series of studies, in the 5-day pilot studies, we exposed animals to between 4 and 12 watts per kilogram. We looked at young and aged animals to see if there was an effect of size on the exposure of RF. And we looked at pregnant rats as well, because our studies, typically we went to design studies starting in utero in our rats. The next phase was the 28-day prechronic toxicology studies, repeat exposure studies, and then the third phase was our 2-year toxicology and carcinogenicity studies. The exposure in these studies were to RFR for approximately 9 hours a day, and the way that we did that was over the course of 18 hours and 20 minutes throughout the day. The exposures were on for 10 minutes, so the animals were being exposed, and then off for 10 minutes. So over the course of those 18 hours and 20 minutes, the animals were exposed for 9 hours and 10 minutes. Rats were exposed either to GSM or CDMA modulated signals at 900 megahertz, as I said, in the rats. These exposures started in utero. In the mice, they were exposed to either GSM or CDMA at 1900 megahertz, beginning at 5 weeks of age. So for the 5-day studies, basically the goal here was to determine whether or not size had an impact on the exposures, on the response. So we looked at 10 different studies. We measured body weight, body temperature, and survival. We didn't look at any histopathology in these animals. We conducted a series, as I said, of 10 studies. We did young animals and aged animals and pregnant rats, each for CDMA and GSM, and we did young and old mice, both for GSM and CDMA. So again, those exposures were 4, 6, 8, 10 watts per kilograms over the course of 5 days. The pregnant rats were exposed from GD10 to GD15. And body temperatures were collected as quickly as possible after the exposures, because we were concerned that as the animal continued to maybe potentially thermoregulate, that as time went on, we would not be able to measure temperatures as accurately. So all temperatures were recorded within 2 to 3 minutes after exposures were ceased. And we took several time points over the course of the 5 days. We considered a body temperature increase of 1 degree C or more to be an upper tolerable exposure limit. That's fairly standard, it seems, for looking at the effects of temperature. So this graph, just to orient you, this is an average of all the time points that we collected over the 5 days for each of the SAR. So you see it at time 0, or at SAR of 0, going up, increasing SAR. We see this graph is looking at aged male mice. This is probably the worst case scenario that we saw in the mice. And we see that there really is no persistent thermal effects observed in mice at SARs up to 12 watts per kilogram. You can see that this is maybe starting to creep up here towards the end of this curve. But there was no effect regardless of the age of the animal, the sex of the animal, or modulation, whether it was GSM or CDMA. In rats, we saw something similar in the young male rats. This is a graph looking at the time points for GSM exposed RFR. We see a very similar thing in the female rats. There is no significant increase, but again, we see as it goes up, it tends to increase as we get towards 12 watts per kilogram. We see something a little different when we look at the old rats, and this kind of indicates that size does matter with the exposures. In the old, heavier rats, we see that there is a significant increase as you increase the SAR, and you see very significant increases at 10 and 12 watts per kilogram. And in fact, 12 watts per kilogram in many of these animals was lethal. So we do see an SAR-dependent increase in the core body temperature following exposure to GSM, as well as a CDMA-modulated RFR. This is in the female rats. We see something very similar in the female rats. The increased SARs above 10 and 12 watts per kilogram seem to be excessive. We did see less pronounced effect in the females as we saw in the males. The body temperatures didn't increase as high. But overall, what we found is that at 10 and 12 watts per kilogram, this was very excessive. We saw increases in body temperature in pregnant rats, in aged rats, in both males and females, and we saw increased mortality in the aged male rats. And as well, when we looked at resorptions in the pregnant rats, we saw that there was an increase in early resorptions at the 12 watts per kilogram in the GSM-modulated exposed animals, but we didn't see that effect in CDMA-exposed animals. At 8 watts per kilogram, we saw that there were several increases of body temperature that were considered excessive. This is over the 1 degree C. This occurred only in the pregnant and aged male rats and aged female rats. And at 6 watts per kilogram, we saw some slight increases in temperature. None of these exceeded the 1 degree C, but we saw these increases only in the aged males and females, not in the young males and females. So we moved on to the 28-day prechronic studies, looking at the exposures of 3, 6, and 9 watts per kilograms in rats, understanding that 9 watts per kilograms, we may actually see a little bit of a heating effect. We wanted to push the exposures as far as we could because we knew that these exposures were one more chance to take a look at what we saw here before we selected the exposures for the 2-year studies. So in the mice, since we didn't see very much of anything, we increased the exposures to 5, 10, and 15 watts per kilograms in the mice to see if we could see something. We were limited as to the power capacity of the exposure system, so 15 was the top end of what we could actually look at without investing a significant amount of money to try to get those exposures higher. In the rats, we had perinatal exposure at 900 megahertz. There were 10 pregnant rats per group, per SAR, per modulation. These exposures began on GD6. And exposures, as before, were 9 hours a day through lactation, and then for an additional 28-day period postnatally. So the animals were exposed from postnatal day 21 to 49 in the rat study. And for the mouse study, animals were exposed 10 animals per group, just a 28-day study starting at 5 weeks of age. And again, I'm not going to present the mouse data, so from this point forward, I'll just be presenting the rat data. So the results from the 28-day study, we saw that there was an increased pup loss during the lactation phase at the 9 watts per kilogram exposure groups exposed to CDMA modulation. We saw decreased body weights in both the dams and pups during this lactation phase. We saw this decreased body weights in the dams at 9 watts per kilogram, and we saw an increase, an SAR-dependent increase in the body weights of male and female pups at both the 6 and 9 watts per kilogram throughout lactation. And although the body weights were lower, what we found is that body weight gains as the study went on were similar to the controls. However, the animals remained up to 17% smaller than the controls through the course of the 28-day study. In this study, we also looked at temperature again to see, again, what we could find at 3, 6, and 9 watts per kilogram. We saw temperature increases in both the GSM-modulated and CDMA-exposed animals at 6 and 9 watts per kilogram in the dams during gestation and lactation in the GSM-modulated exposures. And we saw at 9 watts per kilogram in the CDMA-exposed animals occurring late gestation and lactation. None of these exposures at the 6 watts per kilogram exceeded the 1 degree C that we set as the upper thermal limit. However, several of the time points at the 9 watts per kilogram exceeded the upper limit of temperature increase. And this was not unexpected given what we had seen at the 8 and 10 watts per kilogram in the previous studies. So moving on to the cancer studies, today I'll give kind of an overview of the early data, and then we have complete pathology from the brains and the hearts. So I'll be presenting just the brains and the hearts today. Just to reiterate, this is a complete pathology. We've been through the pathology working group. These are final data for the brains and the hearts. So the study started off, we had 56 time-mated pregnant female HSD rats that were randomly assigned to groups of 1, 1.5, 3, and 6 watts per kilogram. 1.5 watts per kilogram being approximately close to the FCC regulations, which allow a device to expose to 1.6 watts per kilogram to a very local region within the brain. So this is close to what the regulatory limit is. The animals were exposed, again, for the same paradigm of 9 hours a day, through the same on-off cycling, 7 days a week, in utero starting on GD5 and extending through gestation and lactation, at which point we started the clock for the chronic study, at which the animals were exposed to either 13 weeks longer for an interim time point or 107 weeks for the terminal sacrifice. PND21, animals were weaned, the mothers were removed, and animals were group housed until PND35, at which point the animals were individually housed for the remainder of the study. So, perinatal effects, we saw no exposure-related effects on the percentage of dams delivering, the frequency or implantations or resorptions, the number of litters, the litter size, the litter sex distribution of pups, regardless of modulation. Seems like there was no effect of the exposure on any of these parameters. However, what we did see at PND1, we take body weights of the litters, the whole litters, and then just the males in the litters and just the females combined in the litters. And what we found is that there was an SAR-dependent decrease in the mean litter weights of the pups in the males and the females, ranging from about 5 to 8% decrease compared to the controls in the GSM modulated radiofrequency radiation exposures. And we saw in the CDMR exposed animals, we saw a decrease of about 9% just in the female pups. We didn't see it in the male pups as well. So body weights, as we went through lactation, body weights remained a little decreased. We saw decreased body weights in the males and female pups at both 3 and 6 watts per kilogram, ranging from about 5 to 8% decrease in the males and the females. With the CDMA RFR, we saw that the decrease was a little bit more, a little greater in those animals exposed. We saw a decrease in the males of about 10 to 14%, and in the females, 9 to 15% in those pups. So it seems in this case that the CDMA was a little more robust in its response. And in the dams, during that period, we saw that there was about a 7 to 9% decrease in the dam weights at 6 watts per kilogram later on during the lactation phase, during postnatal days 14 to 21, and this was regardless of modulation. So the survival curves from this study indicate that it seems to be that there is a greater survival in the exposure groups, in all of the exposed groups of male rats exposed to GSM RFR. We see that right about here there seems to be a decrease in the males and the controls that we don't see in the females. In the controls that we don't see in the treated, in the male groups. When we look at the females, we see something similar. We see some groups, the survival is a little bit higher. This was the mid group. The 3 watts per kilogram seems to track approximately with the controls, but we do see that in some of those groups there may be a little increase in the survival. When we look at the CDMA exposures in males, we see something fairly similar to what we saw in the GSM. However, we see that there was a decrease through this period of the highest exposures compared to controls. But at the end of the study, we see that all of the exposed groups, the survival is higher than in the control groups. When we look at the females, again, we see something similar to what we saw with the GSM and the CDMA exposed animals, where some of these groups appear to have higher survival at the end of the study, where some of these are similar to the controls. So to move on to the pathology, as I mentioned, we looked specifically at the brain and the heart. So I'll cover the brain first. So these are the hyperplastic lesions that we reserved in the brain. Just to orient you, here is the control. Since it was a common control for both the GSM and CDMA, we've presented the GSM modulation here and the CDMA here. So this is 0, 1.5, 3, and 6 for the GSM. And if you jump over here for the CDMA, what we find in the 90 animals per group, there were no incidences of malignant glioma or the glial cell hyperplasia in the control animals in this study. We compare that to historical controls, and we see that historically there's a rate of about 2% in the studies that we have in the HSD rats this time. When we look at the GSM modulation, we see that there is a small increase in the malignant gliomas in all of the treated groups. This is the incidence and the percent of the animals that are indicated. We also see that there is an increase in, or actually an incidence of the glial cell hyperplasia, which is potentially a preneoplastic lesion that may potentially progress to the malignant gliomas. These are not statistically significant. However, there's a slight increase over controls. When we look at the CDMA modulation, we see that there's an asterisk here indicates that there is a statistically significant trend of 0.003 in the top exposure. And again, we see these glial cell hyperplasias appearing only in the exposed animals, not in the control animals. In female rats, we see, again, we see a couple of incidences of malignant gliomas in some treated animals, as well as the glial cell hyperplasias. There was no statistically significant increase in these brain lesions in the female rats. When it comes to the heart, we observed an increased incidence of schwannomas. Again, there was 0 incidence in the controls for schwannomas or the associated hyperplastic lesion with Schwann cell hyperplasia. Compared to what we see in the exposed groups, we actually see an SAR-dependent, a general SAR-dependent increase here with increasing SAR for the GSM modulation. This is statistically significant by trend test. We see this for both the GSM and the CDMA modulation. As far as the CDMA modulation is concerned, we see a pairwise increase at the highest exposure compared to control, indicating that there's a significant effect here in the CDMA modulation. Again, we see incidences of the Schwann cell hyperplasia in this low exposure group here, as well as the highest exposure group here. And so we see that there overall is a significant difference between the exposed groups and the control groups by poly3 trend test, as well as the pairwise comparison at the highest group of CDMA exposed animals. In female rats, we see similar to what we saw in the brains of the female rats. We see, again, some incidences of these lesions in some of the treatment groups, and again, 0 in the controls. Again, these were not statistically significant when you look at the treated versus the controls. So one of the things that we're interested is in Schwann cells, not just in the heart. It was interesting that we saw these lesions in the heart, but the Schwann cells exist throughout the body. So the pathologists have gone through to look at what was observed for schwannomas throughout the rest of the body. So I've represented these numbers from the 2 slides ago. This is the same numbers that you saw for the heart. What I've added in here is the incidences of schwannomas that was observed in other sites of the body, and what we find is that there really is no increase when you look at the rest of the body that whatever may be going on is specifically occurring in the heart. When you add these lesions together, it dilutes out what we're seeing in the heart, but we still see that there's somewhat of a potential increase, but that's all being driven by the exposures in the heart. And the females, again, these are just the numbers for the females, but we see, again, that we don't really see anything in the females, so there really is no exposure-related change in the incidence of schwannomas in the female rats. So just to summarize from our 2-year studies in the rats, what we've got so far, we see that there's body weights at birth and throughout lactation during exposure in utero tended to be lower than controls. It seemed like the animals that were exposed in utero were smaller. Their growth rates and body weights throughout the course of the study seemed to be similar to controls. This was an effect that was observed early on. In general, we saw that survival was higher or greater in all the groups of GSM or CDMA exposed animals in the male rats compared to the controls. We saw an increased incidence of schwannomas observed in the hearts of male rats at 6 watts per kilogram. This was a significant SIR-dependent trend for both the GSM and CDMA exposure modulations. And there was a significant pairwise increase in the CDMA exposure at 6 watts per kilogram. When we look at the brain, we see that there was a specific SIR-dependent trend in the CDMA-exposed incidence of gliomas in the brain of male rats exposed to RFR, but we do see in general that there was only an incidence of the schwannomas, the gliomas in treated animals and none in the controls. Again, we saw no exposure-related effects observed in the brains or the hearts of female rats. So we concluded from this data that the hyperplastic lesions and the glial cell neoplasms of the heart and the brain observed in the rats are considered likely the result of exposure to whole body GSM or CDMA modulated radiofrequency radiation. There is higher confidence in the results that we observed in the heart than what we observed in the brain, and that exposure of female rats did not affect the incidences of these lesions in the brains or the hearts of the females. So the last thing I want to cover is just quickly, we have genetic tox results. This is going to be, actually I think that this was submitted already, so we are publishing these data, so I will not go in depth into this. I'll just kind of give a quick summary because there was a lot of work that was done here. For the micronucleus, there was no significant increase in the micronucleated red blood cells in rats or mice. But when it came to the Comet assay, we looked at several different subregions within the brain, as well as the liver and the blood, and we found somewhat mixed results depending on the tissue and the brain regions, and depending on whether it was rats and mice. We also saw what seemed to be responders and non-responders, and so this is a graph that I pulled from the manuscript that was just submitted, and this is the incidence of the mean percent tail DNA in control, 1.53 and 6 watts per kilograms. And you can see that there is a significant trend when you look at all the exposures, as well as a significant pairwise increase here in this middle dose group. But what you find is it seems as though there may be responders and non-responders. It seems like there's some very high effect in these two particular animals and these two particular animals, but some of these animals seem to maintain the same levels as what were observed in controls. So overall, I kind of invite you to check out that manuscript when it comes out, but this is kind of the summary of what we see. I'm not going to spend a lot of time on this, but this breaks down, this is rats and mice, males and females. We have CDMA and GSM. So this is somewhat of an adjusted heat map, kind of demonstrating what our results are. If you look at the blocks in yellow, this is where we found statistically significant trend and as well a pairwise significance in one of the exposure groups. In the blue shaded boxes, this is where we saw a significant trend or a particular pairwise significance between one particular group and a control. Green were not significantly significant, but increased in 2 or more of the treatment groups, and everything else we did not see any measurable effect. So just to give you an idea of where we are with the studies and the timeline for completing these studies, The NTP Pathology peer review is underway for the evaluation of all the rat tissues. As I mentioned, we have completed the evaluation of the hearts and the brains. However, we have all the rest of the protocol evaluated tissues that are currently being evaluated for the rats. The mouse study is, I believe, this week. The last of the mouse study materials are being sent, so that will be started in the NTP pathology peer review process as soon as we can get those sorted out. We've shifted resources to accommodate completion of this review as quickly as we possibly can. We're expecting that we should see completed results from this pathology review within the next 18 months or so, and that we'll be working on the NTP technical report concurrently as we're finishing up the peer review process, and we expect that we would be presenting a draft TR to a future public meeting later in 2017 or 2018, and that would be the completion of these studies. With that, I'll take any questions. This is just a slide I put up here because I think each of these are integral part of the conduct of these studies. As I mentioned, we worked with NIST early on to help design this reverberation chamber, as well as come out several times to provide independent monitoring of the exposure systems to get a third-party validation. It is designed the structures and as well maintained the structures, as well as monitored things in real time every day as the studies were going on in Switzerland. And these studies were conducted at ITRI. So there was a close connection between ITRI and it is, as well as bringing NIST into this. So I don't think we could have conducted any of these studies without the cooperation of all four of these groups.
Thank you. Are there any comments for clarification from the board?
I just have a question. From other toxins...
Please identify
Oh,
yourself.
I'm sorry, Stephen Markowitz. From other toxins that may produce gliomas or schwannomas, is it known what the relationship between hyperplasia and the malignancy is?
I think I would defer to the pathologist, but I know in the peer review process, there was a lot of questions as far as the gliomas and the difference between what was observed as the glial cell hyperplasia versus malignant glioma. And I believe that what they saw was that the only difference really between them was not a cellularity or any kind of other issue, was just basically size. And so really it seems like one of the main differences between the glial hyperplasias and the malignant gliomas was really just size. And when you look at those numbers, there are different numbers. If an animal was diagnosed with the hyperplasia, if it was diagnosed with a glioma, it was not diagnosed with hyperplasia. So those numbers would be additive if you were to consider those hyperplasias to be small gliomas.
Cindy Afshari. I had a question. At the end, you talked about having a lower degree of confidence in terms of the observation of the gliomas. I just wanted to clarify if that was because of the fact that the incidence is occurring at the same level as the historical controls, and then the fact that the survival was lower in the controls in this group. Is that calling into question your trend test that you were referring to? Could you just clarify your lack of confidence and where that's stemming from?
No, the statistics take into account the survivability, but really the confidence comes from the level of statistical significance. We see that there's more statistical significance with the heart. We just don't see the level in the brain that we saw with the heart. So it's more of our confidence, not the statistical confidence.
And if I can, one other question. When you talked about the decreased body weight in the beginning, I don't know if you can draw any comparisons in terms of ionizing radiation or other similar studies in terms of how that compares.
Yeah, I'm not sure how that would compare, and that's one of the things that we're looking into. What may be occurring at the early on stages where we see decreased body weights. Again, like I mentioned, throughout the course of the study, they seem to gain weight just like the others, and there doesn't seem to be any kind of difference. That seems to be an effect that occurs early on during lactation, and from birth through lactation.
Dr. Ramos?
Yeah, Ken Ramos here. First a comment and then a question. I actually would rethink carefully the answer that you provided with regards to gliomas versus glial hyperplasia, because I actually think that it's not correct. Those two things are two different entities, so don't. The only possible connection that you would have in there is the idea that perhaps the hyperplasia would somehow set the stage for occurrence of malignancy. But even that's, I think, contentious. So don't treat those two things as something that's comparable. I would rethink that answer very carefully. The question actually is, how did you use the historical control data in interpreting the data that you came up with? To me, it seemed that it was not used in interpretation. And then the question becomes, why did you include it in the slide, and why are you mindful of that historical data?
Well, to answer your first question, I would defer to the pathologist on that, and that was a very contentious issue as far as what was hyperplasia and glioma. On the second question, the historical controls are looked at. In toxicology, we consider the concurrent controls to be the best controls. In this case, we know that those controls were handled the exact same way. We know that in this case, the animals were individually housed. In most of those other studies, I think only one of those other studies, they were individually housed. We know historically from Fisher rats that the individually housed animals, their survivals are much lower. As far as looking at the historical controls, we incorporate that into our evaluation. We looked at the percentage to see how low the background rates are, how rare of a tumor this was. And so we did actually look at those historical controls and evaluated that as well, and that we incorporated that into our conclusions.
It seems to me that you probably ought to then go back and kind of relook at the data, because I only glanced, of course, I was just moving through the slides, but it would seem to me that if you do take into account historical controls, then your interpretation of some of the findings is going to have to be changed, because then that means that some of the signal that you were seeing in the results might actually be noise. And one of the comments that I actually wrote in here is that, and it's only a sense of what I'm reacting to. Obviously, you know your data better than I would. When I looked at your results, I get the sense that you're getting drifts in that data set that somehow you're really not able to kind of pin down and explain. Your data, your weight data actually kind of hints to that. There is something going on that you're really not picking up. And tumor occurrence in itself actually gives you a little bit of noise that if you were to use historical controls, in some instances would actually negate some of the conclusions that you put down. So I would just kind of go back and maybe rethink that piece.
So I'd like to comment a moment on the historical control, which, over the years we always look at the historical control, but as Michael said, the concurrent control is always the best control. In this case, the Harlan-Worcester rat, or the Harlan-Worcester, I think, Harlan Sprague-Dawlian rat, we only have, I think it's 10 or 11 at most, other studies using this rat, none at this laboratory, all done under other conditions, different housing, and so on. So which really makes the use of the historical control it's almost, they're not really historical controls, they're controls from a limited number of other studies done at other laboratories under totally different conditions than were in this study. So that's something to think about. We did look at it. We did look at the totality. You still have the schwannoma is unequivocally clear. You don't see it in any of the others. The gliomas are, in fact, because in one or two of the other studies, there was a high level of glioma. I don't know if Michael, probably not the time to comment now. We are going in and doing molecular genetic studies, trying to look at the genomic sequences that are present in when gliomas appeared in some of the other studies that use this strain of rats, try to get a molecular signature there and compare it to the molecular signature in the gliomas in this study. But that you'll have to wait for the data because it's still very much in the process of being generated. And John wanted to comment on your comment, Ken, about that the glial hyperplasia and the gliomas are not, in fact, In fact, the pathology reviews that have been done by veterinary neuropathologists very much believe that these are essentially the same lesion. John?
Yeah, what was I going to say that? So obviously,
you can say it better than I can.
No, probably not. So clearly, when we got this information in from the laboratory, we looked at it. We have had many people, many eyes look at this information, and overall, the conclusion is that you come down to at the end of the day, even taking into consideration all of the information on historical data, the same kind of things that Linda talked about. And you come down to the end of the day, is it more likely that it's related, or is it more likely that it's not related? And the majority of people, and I will say it's not uniform, it's not unanimous. The majority of the people that look at this data set say it's more likely that it is related, that these, certainly the schwannomas, and we have less confidence in the gliomas, they are related to radiofrequency radiation more so than they're not related to radiofrequency radiation. So that's sort of the line at which we've come down on. With respect to the relationship of hyperplasia and the gliomas, hyperplasias, and the schwannomas and the hyperplasias, we have convened, I believe it's 5 separate pathology review groups to look at these lesions. 2 of the groups were charged with the specific question that you raised, which is there a relationship between these two lesion types? And that Michael's correct in that the conclusion was that they are related, that the only difference in these hyperplastic lesions is size, and that they would be considered potentially gliomas if they were to progress. So that's where we were with that. But we took that question to the best experts we could find, and that was their opinion.
Well, that's helpful information. I would still maybe continue to seek additional sort of insight into that, because in my view, the interpretation that's been rendered is contrary to what I have heard, neuro-oncologists, articulate. That said, going back to the issue of historical controls, in light of what Linda said, I would actually then not even make reference to it, because they're not of any value to you in your interpretation of the findings that you have for the reasons that Linda articulated. So in essence, what they do is cloud the issue. If you sort of kind of have that as a backdrop to what you're trying to say, then the last point that I would make is that, and it relates back to this sort of sense of some sort of noise signal that you've got in there. If you notice, when you say that you have more confidence in the schwannomas and in the gliomas, all that goes back really to what your control data was. Because if you look, you actually have higher incidence of those tumors in your control population relative to your treated. And that's what sort of probably is giving you that quote level of confidence.
I should say that with the schwannomas as well, there is a beautiful dose-response relationship that is not as clear in the gliomas.
Yes.
Yeah, I thought I saw a comment over on this side. George, or did you change your mind? Okay, go ahead. Well, we can come back to you.
I may return to this at a later point, but the brain lesions, none of them reached a level of statistical significance. Is that correct or incorrect?
The CDMA exposure for the malignant gliomas was a statistical trend.
At the 6 watt?
Were for all of the exposures.
It's just trend analysis.
Pairwise comparison was not statistically significant.
Okay, thanks.
Go ahead, one last question.
So if the glial hyperplasia and glioma is a clear continuum, very closely related, then why can't you add them together, which gives you more statistical power? I'm sure you've done that. I'm just asking you what your thought about that is.
And maybe the pathologist would care to comment on this, but the fact that they have delineated size the difference in hyperplasias and neoplasias in other organ systems is also based on size. For example, in the kidney, the involvement of various tubules, a couple of tubules is a hyperplasia, a few more tubules is an adenoma. So, I mean, this is something that is typically seen in veterinary pathology. I suspect it's also seen in human pathology to some degree, but it's not unusual. And where the pathologists care to draw the line, and they come to an agreement on that line, we stick to that line.
Okay, thank you very much. We're going to move on.